Lecture 2 relates to the last part of Chapter 3, which talks about lipids, proteins, and nucleic acids. So concept 3.4, we're going to talk about lipids. Lipids are what are called hydrophobic. Hydro relates to water, phobic means fearing. So these molecules actually are repelled by water, and you can see that if you mix oil and water, where the water kind of pushes oil out of the way and is attracted to other water molecules and then separates from the oil. That is because water actually has a charge to it. If you look at the picture, the oxygen, which is this larger molecule, ends up having a bit of a negative charge to it. The hydrogens, which are a smaller molecule, end up having a bit of a positive charge to it because the oxygen, which is larger, that atom actually draws and pulls the hydrogen's electrons more closely to it. Those electrons have a negative charge. So that makes the oxygen end more negative and the hydrogen end more positive. So it's like a magnet. So polar molecules are like a magnet with a negative and a positive charge on each end, basically. Because of this, water molecules attract to each other. So this is how they can displace oil or push it out of the way, because now these negative sides are going to be more attracted to the positive sides of those polar molecules. So lipids are hydrophobic. They do not have a charge. They don't have positive and negative sides. They also are not true polymers in that they don't chain up the way carbohydrates do. We saw, monosaccharides chaining up to form polysaccharides. Lipids don't do that, and I'll describe that a little bit more in the next slide. They are hydrophobic, meaning they fear water or are kind of repelled by water because they are nonpolar. The hydrocarbon just means that they have a lot of hydrogen and carbons. Again, that the carbon forms the backbone, and then those carbons are connected to hydrogens. Examples includes fats, phospholipids, and steroids. We'll talk about fats as being a part of our diet as a triglyceride. Phospholipids are in our cell membranes and are a lipid bilayer. There's two layers of the phospholipid molecule. And then steroids are another example of a lipid. And we'll talk a little bit more specifically about cholesterol, which is a type of steroid. The polar water molecule then has a charge. It is considered to be hydrophilic, meaning it likes water. Those hydrogen bonds then connect between the negative oxygen and the positive hydrogen. So this would be that hydrogen bond, which then repels oils or nonpolar liquids, so that we have water molecules connecting to each other, meaning they exclude fats and connect to each other. Fats are the main energy storage. We go to carbs for our energy typically. We store fats. If you're on a diet, say, and you're not eating as many carbs, then you can tap into those stored fats for energy, which is how we lose weight. Fats typically contain two types of molecules, I should say. There's a glycerol, which is a 3-carbon alcohol, and that would be the glycerol molecule with a 3-carbon backbone. And then we connect the glycerol to fatty acids. And in the case of a triglycerol, this would be typical of the fats that we eat. There are 3 fatty acid tails hanging off the one glycerol molecule. And in the last slide, we talked about fat not being a true polymer, or lipid, I should say, not being a true polymer, because this is how it arranges itself. The fatty acids don't chain up one right after the other. You really just have the fatty acid itself chaining to the glycerol, which makes this this molecule that's almost in the shape of a letter E. So fatty acid types, when we make triglycerides, we can make them with fatty acids that are either saturated or unsaturated. Saturated fats have the maximum number of hydrogen atoms, and there are no double bonds. This causes the chain to be a very straight type of a fatty acid chain, meaning the chains connect very closely together or sit side by side in a very close relationship. And that actually is why they are solid at room temperature, because those fatty acid chains are very straight and thus sit next to each other very closely. On the other hand, unsaturated fats have double bonds periodically, So they do have double bonds. The double bond gives it a little bit of a kink so that the fatty acid tails can't sit so closely together. As a result, these fats end up to be liquid at room temperature. So saturated fats tend to be animal fats and are solid at room temp. Unsaturated fats with the double bonds tend to be plant and fish fats, which are liquid at room temp. A little bit more healthy for us also. So phospholipids are another type of lipid. These include a molecule containing only two fatty acid tails along with a phosphate group. The phosphate group is a charged molecule, so this means it is polar, which then means it's going to have a charge to it again. It's like a magnet. So this side of it is going to connect to the water portions within living things, and as such, it sets up this cell membrane that we see, where you have the polar head, on both the kind of this. If this is external, and this is internal, this might be the cytoplasm. We're going to see that polar head closest to the water on the outside of the cell, and then the second polar head closest to the water on the inside of the cell because of that charge. The fatty acid tails are hydrophobic again, and they form the inner part of the membrane, kind of repelled by water. And because of this configuration where we have the polar and then the nonpolar, that's how we set up cell membranes, and all membranes contain this molecule. It is in a bilayer, meaning there are two layers of that molecule, and the polar end again is going to point to both the exterior and the interior of the cell, which then the tails are going to hide out in the middle. So this is the structure again that forms the main structure of cell membranes and forms boundaries of cells, because this molecule kind of sets itself up and defines the inside and the outside of the cell. Steroids would be another example of a lipid. One example of that would be cholesterol. The difference with steroids is there's one fatty acid tail with these 4 fused carbon molecules or rings. Cholesterol is also found in animal cell membranes, and one of the functions of cholesterol is really to keep those membranes a little bit more separate, and it allows membranes to function better in temperature different situations, meaning where it's colder or warmer, it allows the membrane to be a little bit more flexible. And we'll talk more about this in chapter 7 actually. But cholesterol would be a type of steroid, which is a type of lipid, and its configuration or its form is where it has one fatty acid tail with these 4 fused carbon rings. The problem with cholesterol, we make it ourselves, but when we add it in our diet, it then can end up in our blood vessels, and it can, with high blood levels, this can lead to cardiovascular disease because the cholesterol adheres to the sides of the vessels and can cause those vessels to plug up, and that can lead to high blood pressure or cardiovascular issues. So proteins are the next type of molecule we'll talk a little bit about. Proteins are different from lipids and carbs in that they don't provide energy for us, they really provide structure. It really is the structure that makes up most living bodies. For example, our muscle, our bones, our skin, our hair, our fingernails, those are all protein molecules. So we eat protein in order to build body mass. Several functions. They help us protect us from invading organisms. We have white blood cells that can ingest bacteria, etc. if we get some kind of an infection, and those white blood cells are mainly protein. Storage. We're able to store calcium in muscle cells. We store calcium in bones. We store sodium, potassium, so we're able to store things in these molecules. We can transport things with these molecules. There are proteins in cell membranes that allow certain molecules to be transported in and out of the cell. Hemoglobin is a molecule in blood cells that transports oxygen, so proteins help us move things around the body. Cellular communication. There are other proteins in the membrane that connect to hormones, which signal different things going on both in and out of the cell. So it allows for communication between the inside and the outside of the cell. as well as inside the cell. So we can communicate with these proteins. Movement, muscles move bones, so those types of proteins are able to move things. And then again, they provide most of the structural support for the living organism. So lots of different functions. Proteins are made up of things called polypeptides. And polypeptides are really just polymers built from amino acids. There are 20 total amino acids, and when we chain up those amino acids in various ways, we then can build these polypeptides, which make up protein. And most protein consists of one or more polypeptides. So again, when we talk about proteins, we're really talking about structure. So hair, skin, muscles, ligaments. The body parts are mostly protein, and so we say that protein leads to the structural mass of most cells, along and has many different functions. This slide just kind of talks a little bit more about the different types of functions that proteins have. So we've got storage proteins that are going to store various things. We have defensive proteins that protect. We have transport proteins that move things in and out of the cell. We have receptors proteins that connect to things and then signal information to both the inside of the cell and the outside of the cell. We have contractile proteins that help things move. Our muscles move our bones, our skeletal muscles move our bones, etc. So these are all just the various functions of proteins. With proteins, some of these protein molecules are called enzymes. Enzymes are catalysts that speed up reactions. A lot of different chemical reactions going on in the body. All of those reactions need enzymes to properly function because enzymes are those things that speed up those reactions. Enzymes are not used up. They're reusable. And their job is to do something. They're very specific. So what we see, if this is the enzyme is the thing that's going to do the work. The substrate is the thing that's going to be worked on. In this case, we are taking this larger protein or substance, and we are going to break it down with the help of that enzyme. So the enzyme is a molecule that has a certain function, and once it does that function, it can just go back and do the function again. It's not used up. So it helps us speed up these chemical reactions that need to take place in our body. For example, we have to break down food that we take in our mouth, and we have lots of different enzymes to help us do that. Those enzymes, such as a disaccharide, which is an enzyme that's going to break down disaccharides, which are a 2-sugar molecule. That disaccharide can break down one disaccharide molecule and then can go back and break down lots more. So they help us perform these chemical reactions throughout the body. Those are called enzymatic proteins. Amino acids are the building blocks for proteins. This slide just goes into the different types. There are 20 different types, and they differ because of the side chain, this yellow side chain that you see. Those side chains are called an R group. They have the same basic structure in all amino acids, but it is the R group that is different. You'll notice some of these side group chains are nonpolar, I should say, or hydrophobic in the yellow, and then we do have some that are polar. But it is basically the difference of those R groups that give us the 20 different amino acids, and the combination of those 20 different amino acids gives us all the different type of protein we see, whether it's bone or muscle or hair or fingernail or the cornea of your eye. Polypeptides are what forms the protein. These are chains of amino acids. So these amino acids link with something called a peptide bond. This would be that peptide bond, and by linking those amino acid groups together with these peptide bonds, we call them polypeptide polymers, where we're chaining this up, and we have multiple amino acids depending upon the type of protein you're talking about. Protein structure really relates to its function. So, we have, this would be a type of protein, and we've got this kinked and coiled and multi-stranded structure. So each protein has one or more polypeptides that twists and folds and coils into a unique shape. And its shape is usually 3D. It's a large molecule. It is determined genetically, and that genetic information then leads to the sequence of amino acids, and each protein has a different sequence of amino acids and typically a different shape. Its shape and structure really relates to its function. So, for example, a hemoglobin molecule is kind of circular. It's in red blood cells, which are also circular, and it helps us transport oxygen around the body. So, typically, when you see the structure of something, it often leads to what its function is, or can relate to what its function is. This picture just shows two different types of proteins. We've got an antibody type of protein and then a protein in the flu virus, and you notice they kind of go very closely, neatly fit together. So typically, proteins are very specific for what their function is, where their shape really relates to what they're going to act on. There's basically 4 levels to the making of a protein. What that means is there's 4 steps to the building of it, and they lead to kind of the structure within that step. So primarily, there's a unique amino acid sequence, and that would be right here, where you have just this sequence of amino acids that is genetically determined. These amino acids connect with those peptide bonds. So we would say that this would be a polypeptide structure with those amino acids linking up in a specific way. Secondarily, this coils and kind of folds upon itself. And so now we say this polypeptide chain has the secondary structure, which is kind of coiled and folded. Once we get that coiling and folding, the tertiary structure kind of sets itself up. This is where the side chains interact. Remember, some are going to be nonpolar, some are going to be polar, and so some are attracted, some are repelled, and that is what actually gives it a little bit of that 3D shape. The quaternary level is where now we're going to interact with multiple polypeptides. So here we have several different polypeptide chains coming together, and this would be the ultimate final protein, which is made up of these multiple polypeptide chains. The spider web really is just an example of a type of protein, and that spider is producing that silk, which then is formed into the spider web shape. Collagen is another type of protein where we have these multiple polypeptide chains coming together, interacting in a folded, kind of interwoven form. Hemoglobin, I mentioned earlier, this is the protein that is within red blood cells and helps us carry oxygen. It includes an iron group, which is connected to that heme or portion of the polypeptide. So here you have multiple subunits coming together to form this hemoglobin molecule, which is found in blood cells and helps us carry oxygen. Sickle cell disease would be an example of where there's a change in the structure. So with that change, we have the normal structure here. The sickle cell structure is where there's a change in the amino acid sequence. It is just one amino acid that's different. The valine replaces the glutamine. And with that change, then we have a different shape. It's no longer circular. And as a result of this different shape now, we get the blood cell looking a little bit different. So the genetic disorder sickle cell disease is where a single amino acid substitution, meaning one replace the other, affects the structure of hemoglobin, which then affects its function. This does not move through the blood vessel the way it should. And so these people have what's called anemia, where they often are very weak and fatigued. It's very painful. So kind of an awful disease, and it's based on just one amino acid substituting with another. So the structure of protein is really determined genetically, but we can use a tool called X-ray crystallography in order to see that 3D structure. Sometimes we see that the structure is altered. Some conditions can cause proteins to unravel and then they don't function properly. So pH, for example, that's the level of acidity. For example, sometimes if you have really acidic food, that lowers the pH of your stomach so much that the enzymes don't function properly. They are denatured a little bit, and then you have a harder time digesting that food, and that can lead to an upset stomach. Salt concentration is another. Temperature also can affect if it's really cold outside. The enzymes in our muscles don't work properly because now it's so cold, so your fingers kind of feel like they don't move as well as they should. So these are factors, including other environmental factors, that really just cause the protein shape to unravel a bit so they're not going to function as properly as they should. If this happens to a certain degree, we call that protein denatured, where you can actually render it biologically inactive because it's changed its structure so much. With X-ray crystallography, then, it gives us a picture of that protein and we can tell if the shape is the way it should be or not. Concept 3.6, this is the last section of the chapter, really talks a little bit about nucleic acids. Those are molecules that store, transmit, and express hereditary information. All that means is our external features, eye color, hair color, skin color, etc., are based on these nucleic acids, which are passed on in a hereditary fashion and give us the traits that make up our characteristics. And all life forms have this genetic material that determines what their external form and shape and function is going to be. There are two types of these molecules. One is called DNA. Long form of that word is deoxyribonucleic acid. This is the genetic material which contains the genes which determine the traits. These are an example of a nucleic acid, and this nucleic acid is made up of monomer units or single units called nucleotides, and we'll talk a little bit more about what the shape of that is in upcoming slides. They are polynucleotides, meaning they have lots of these nucleotides strung together. So again, we're talking about kind of the chaining up of these nucleotides, which then makes this DNA kind of look the way it does and then function the way it does. Ribonucleic acid is a second type of nucleic acid. This is really the molecule that's building the proteins, but it builds the proteins based on the code from the DNA. The DNA is then that genetic material that provides the blueprint, and then the ribonucleic acid would be the molecule that's actually going to build that genetic material or that protein structure based on the genetic code. DNA directs this molecule and controls what the RNA is producing. When we talk about protein synthesis or the RNA building a molecule, if we end up with a polypeptide, this code really comes from the DNA molecule. And we've got to get that code to the ribosome, which is where protein is built. So the function of ribosomes is to build protein. The code for that is back in the DNA molecule. So this slide just shows you the process of going from DNA to mRNA, which is the messenger RNA molecule that has a copy of the code from the DNA. This messenger RNA now is going to move to the ribosome, which is going to be the site where that protein is actually built. So this process is called transcription translation. We'll talk a little bit more about this in later chapters, but this is the process by which we get that information from the DNA molecule, which contains the genes that code for the building of this protein molecule. So nucleotides then are, either the genetic or the protein building material. They're made up of what's called nucleotides. We're going to say that those are the subunits, and the nucleotide makes up both DNA and RNA. They look a little bit different between those two molecules, but basically the same type of structure. A nucleotide contains three parts. It contains a nitrogenous base, and that really is just a base that contains nitrogen. This would be that nitrogenous base. Contains a pentose sugar, that means it is a 5-carbon sugar right here. and then it contains a phosphate group right here. So it's the combination of those three things which make up the nucleotide. It is the nitrogenous base that changes. These are the different nitrogenous bases. We have things called purines, which are a single ring structure. It's a longer word, uh, but it contains a smaller molecule, which is a one ring structure. And the three types of pyrimidine, include cytosine, thymine, and uracil. You'll notice that thymine is in DNA, uracil is in RNA. If we look at a second type of nitrogenous base, these are the purines. You'll notice they're bigger. They have two ringed structure molecules associated with these types. Purines is a smaller word, but it's a larger molecule. The two types of purines include adenine and guanine. And we see those in both DNA and RNA. It's really just the thymine and the uracil that are found in different molecules. Thymine will only be in DNA, uracil will only be in RNA. But these 5 different types of nitrogenous bases then would be this part of the structure that changes. The phosphate and the sugar groups remain the same throughout both. You'll notice the phosphate and the sugar make up the rung of the ladder, and then these nitrogenous bases hang off the sides. Either it's the 2 ring structure in the purine or the 1 ring structure in the pyrimidine. But getting back to the information, we've got DNA has a sugar deoxyribose. It is still a 5-carbon sugar, but we're going to call it a deoxyribose, which is where the D comes from. RNA contains ribose, which is where the R comes from. Again, that would be that sugar unit, a little bit different between DNA and RNA, still a 5-carbon sugar, and will still make up the backbone of the molecule. So the backbone is made up of the sugar and phosphate units, whether it's the deoxyribose or the ribose in DNA and RNA. So that is consistent between the two, but then those nitrogenous bases hang off as appendages, and it is the changing of these bases which determines the genetic sequence and what that structure is going to end up to be. And we also have a little bit of a difference between the nitrogenous bases in RNA and DNA, where you're going to see T's in DNA for thymine, you're going to see U's in RNA for uracil. So here we've got a bit of a typo. It really should be a one-ring structure. So with the with the pyrimidine. So again, this is a longer word, but a smaller molecule. This includes cytosine, often abbreviated with a C, often abbreviated with a T, and uracil abbreviated with a U. Again, we're going to see the thymine only in DNA and the uracil only in RNA. But those would be smaller molecules, one-ringed, and we're going to call them with this larger word the pyrimidine. One way to remember it. The purines have a 2-ring structure. So adenine and guanine, abbreviated AG, are larger molecules with this smaller word as their descriptor. We see that down here again. Cytosine and thymine and uracil, 1-ring structure, and we're going to call them pyrimidines. Purines include this 2-ring structure, adenine and guanine, a little larger, but this smaller word describes them. So we will only find thymine in DNA, we will only see urine and RNA, the others are in both molecules. Because of their structure, the 1 ring and the 2 ring, we see them pairing up in a certain way. With DNA, we have a spiral double helix. That means it's 2 sides. So the sides of the ladder will be the sugar and the phosphate, and that kind of repeats itself over and over the whole ring. The rungs of the ladder are bases. The width of this space between the 2 sides, there's only room for 3 bases. And that's important because with DNA, you can only get a certain pairing with this, in that you can only have 1 pyrimidine and then 1 purine. You could never have 2 pyrimidines because that would be too small. Remember, those are the smaller one ring structure, and you'd end up with just two rings. You couldn't have two purines because those are the two ring structures. That would be four ring width. That's too wide. I hope this makes sense. But DNA, the nucleotides, I should say, the nitrogenous bases arrange themselves so that there's a width of three molecules. With that, then, in DNA, A will only go with T. Remember, adenine is a two-ring structure. It's a purine. Thymine is a one-ring structure. It is a pyrimidine. So then we have three rings across. Guanine will only pair with cytosine. Again, it's that same scenario. Why the two? Why is it adenine with thymine and not with guanine? Evolution, our evolution has kind of determined that, but it always ends up that it is A with T always in DNA, G with C always in DNA. In RNA, again, we're going to see some of the same nitrogenous bases. This is a single polypeptide chain, but they still pair up the same way, where you have A with U always. Remember, we're replacing the T with U in RNA, and G again will always go with C. So we call this complementary base pairing, and the next picture shows you a little bit more about that. So if we look at this picture, you'll notice that guanine is longer. It is a purine with two rings. Cytosine is a pyrimidine with one ring, and they fit between the spaces of the ladder, of the rungs, the rungs, I should say, fit between the sides of the ladder. The sides, you can't see it, but here, this would be the sugar and the phosphate kind of repeating itself. You'll notice the A as a purine is longer, the T as a pyrimidine is shorter. So because of this configuration of only having room for three bases, this leads to that complementary pairing of A with T, always in DNA, G with C, always. You could say it the flipped way. T will always go with A, C will always go with G. We see the same thing here, where we've got G and C pairing up together. In RNA, though, now, we are going to have the U instead of the T pairing up with the A. With DNA and proteins, then, evolution has determined a lot of this. We inherit these nucleotides, so then our body shape, again, determined by the DNA, goes along with the nucleotides that we inherit and the sequence of those nucleotides. With molecular biology, we'll talk more about this when we get to evolution, but you can kind of determine how closely related organisms are by their structure, how close together, or how similar that molecular biology is. So then we can look at these sequences of amino acids, and it shows that, it's pretty similar, correct? Where everything is pretty much the same between the human, the monkey, and the Gibson for this amino acid sequence for hemoglobin, or the beta globin, I should say. But the more closely related that molecular biology, the more closely related the species is because of the similar DNA. And that has to do with our evolution, where everything is connected. The more closely related, what that means is the more recent our common ancestor, the more different we are. That means the more distant our common ancestor. The last slide kind of goes into just a review of all the different biological molecules. So again, we've got kind of this summary of carbs. It shows you that monosaccharides would be that monomer unit. These are different examples of carbohydrates. And then this would be their function. Main function is energy. It's our fuel. It's what we use for fuel. But it can also be, it's in the cell walls. We do store some, and it is also a part of fungal cell walls as well. And so there's several functions for carbs, but mainly it is our fuel source. It's our immediate fuel source. Lipids, then we have these three different types, where we have the triglyceride, the phospholipid, and the steroids. This is going to be a main stored energy source. It is also found in cell membranes, and we can, it also helps some signaling molecules, sorry, excuse me, and but basically in cell membranes, and it's a stored energy source. Proteins are going to be those molecules that provide structure. They tend to have amino acids, which are connected by these peptide bonds with that R chain, which then allows them to kind of interact with each other. Lots of different functions for proteins, and lots of different examples as well. And then lastly, nucleic acids express that heredity information, allow us to build proteins according to that energy or that inherited information. We've got this main structure, the nucleotide, which contains the nitrogenous base, a sugar, and a phosphate group. The two types would be DNA and RNA. DNA is a double-sided molecule with the nitrogenous bases cytosine, guanine, adenine, thymine. RNA is a single sided molecule with the nitrogenous bases cytosine, guanine, adenine, uracil. DNA stores hereditary information, and RNA functions in gene expression. And what that means is it carries out and builds the protein according to the DNA directions. Thank you. Have a great day.